Hasib Rahman Shah, and today we will discuss how real-time PCR works, what are its principles and different modules. In real-time PCR, the reaction is monitored in real time by linking the amplification of DNA to the generation of fluorescence, which can simply be detected with a detector during each PCR cycle. It means that real-time PCR, we can see our PCR process, that whether it is happening or not. If our DNA is amplified or is going to amplified, we will see it on the screen, so we can monitor our DNA amplification at real time. So that's why it is called real-time PCR. Detection chemistry, how we detect it at whole time when it is happening. When designing a real-time PCR experiment, a significant decision is choosing the correct detection chemistry for your application. There are two major options to choose from: intercalating dye, hydrolysis probe. So what is intercalating dye? We will discuss it first, and then hydrolyzing probe. The dye is fluorescent in its own right, but in the presence of double-stranded DNA, the dye intercalates with the DNA double helix. This alters the structure of the dye and causes it to fluoresce more. So very simple, as a PCR creates more DNA, more dye can bind and more fluorescence is generated. The most commonly used dye is cyber green. Dye in solution emits low fluorescence, while on my right, if you can see, emission of the fluorescence by binding to DNA produces more fluorescence. So we can get an idea in real time when there is PCR going on. Hydrolysis probes. Probes are fluorescently labeled DNA oligonucleotides. They are designed to bind downstream of one of the primers during the PCR reaction and to give a fluorescent signal during the reaction. The 5' end of the probe is labeled with a fluorescent reporter molecule. The 3' end of the probe is a quencher molecule. This is another molecule that effectively quench, means it absorb the light or energy which is produced by fluorescent reporter molecules. So we will see no light or fluorescence. The output from the reporter. Therefore, when the reporter and quencher are physically close to one another, the overall level of fluorescent output is low. What is TaqMan Probe? When intact, the fluorescence of the reporter is quenched due to its proximity to quencher, as I explained earlier. Probe hybridizes to the target. So what happened when probe hybridizes to the target molecule? Then double-stranded DNA specific 5' to 3' exonuclease activity of Taq cleaves off the reporter. We will see in next slide as pictorial, which will remove your confusion, if any. Reporter is separated from the quencher and a fluorescent signal is generated. Signal is proportional to the amount of amplified product in the sample. Molecular beacons. Probe sequence before binding or before annealing to the target DNA molecule. Molecular beacons are hairpin structures composed of 25 to 40 nucleotide base paired stem and a target specific nucleotide loop. The loop consists of target specific nucleotide probe sequences 15 to 30 nucleotide. A fluorescent moiety reporter is attached to 5' end and a quencher moiety is attached to 3' end. The stem keeps both the moieties in close proximity so that fluorescence is quenched. Molecular beacon is non-fluorescent due to close proximity of non-fluorescent quencher and the fluorescent reporter. The probe denatures and the loop anneals to the target sequence of the amplicon, separating the quencher from the fluorophore and thereby producing fluorescence, which is proportional to the amplicons produced during PCR. Molecular beacon is displaced, not destroyed, during amplification because a DNA polymerase lacking 5' exonuclease activity is used. So here it doesn't lost. Process diagram. You can see that after denaturation, probe bind to the target DNA molecule. So TaqMan polymerase, what it does, it remove the fluorescent molecule from the probe, but because it has 5' to 3' exonuclease activity. But in TaqMan probe, the 5' exonucleic activity is not present, so just the probe is amplified, which causes quencher and fluorophore a distance between them, a long distance, which causes fluorescence. Real-time PCR analysis in exponential phase. In non-exponential phase, we can not see CQ value non-exponential plating. So this is the analysis of PCR, and that's all. So thank you so much for watching, and please, if you liked the video, please subscribe my channel and like it, comment, and I will improve videos. And if you have any problem in PCR or in biotechnology related, so I will make videos for you if you demand in comments. So please guys, comment. Thank you so much. Bye.